Thank you all for coming. Welcome to the last session of the ICAL Seminar Series 2024-25. We are very pleased to wrap up the year with Alberto, who is a former alumni from Imperial College London, and who is senior staff engineer at InstaDeep, and is going to be telling us about their work with foundational biological models. So for those of you who are new to this, after the talk, there will be some time to network and make further questions if you want. The networking session as well is sponsored together with the seminars by our sponsor InstaDeep, DeepMind, and Iconic. And if you like the series, just for you to know, we will be coming back in September with further speakers and topics to cover. Now, with all that said, Alberto, the stage is yours. Thank you for coming.
Thank you very much. Thank you very much for inviting me today. I will indeed talk about foundation models in biology. So I am an applied researcher at InstaDeep, the AI subsidiary of the company that made the first COVID vaccine, BioNTech. BioNTech's mission is to develop immunotherapies and mRNA vaccines to cure all sorts of diseases, notably cancer. And what I want to show you today is the kind of AI models that are used in modern research towards curing these diseases. So after a brief introduction to our company, we will be talking about foundation models. But before we talk about foundation models, I would like to share some foundations in biology, in case you are not that familiar with the topic, and so that we can have a common language later. After that, we'll dive into some applications that have been developed at InstaDeep. So briefly about the company. InstaDeep has a diverse set of partners with whom we build strong relationships. With our partners, our mission is to push AI to the benefit of everybody, and that has been recognized many times. To do that, we have to find talents from across the globe. Concretely, that means we work in a multicultural environment, which is very enriching, but we also have lots of resources available to conduct our work efficiently. But really, our best resource is our people. What is it that we do? We do three things. We conduct world-leading AI research, deliver groundbreaking AI solutions, and we offer a diverse expertise across a wide range of different industries. This has been successful, so we have top-tier customers for whom we deliver actual solutions. By actual solutions, I mean that we build products for these customers that actually solve real-world problems. And in order to build these products, we need domain experts. So this means that at InstaDeep, we have people who know about a wide range of different things. So for example, we have people who work in applying AI to the field of scheduling, well, to the application of scheduling trains with AI methodologies. We also have people who work, for example, on circuit board designs, which is incredibly complex. But what I'm here to talk about today is biology and foundation models applied to biology. So InstaDeep is characterized by this marriage between research and applied teams. And that's why I'm here today, perhaps a little bit unusually, not to talk about my own research, but rather the research that surrounds me, developed by my colleagues in the research teams. And also, as a party to this marriage, I can honestly say, what a marriage. We're very lucky to have fabulous researchers publishing in top journals and very prestigious conferences, and they do so across various disciplines. So I'm here to talk about biology. So here is a selection of foundation model of our latest papers in this field, but we have just as prestigious a list across other disciplines as well. Now, just a final word on the company. I think 2023 may be the year in which you first heard about InstaDeep, if you hadn't heard of it before, because that's the year when we were acquired by BioNTech. BioNTech, as I was saying, is a leading pharmaceutical company, and it's aiming to do very ambitious things, like cure all types of cancer. So as an engineer collaborating with their wet lab teams, I get to work both in AI and with wet labs to solve real-world problems in this very exciting industry. This experience of collaborating with wet lab teams has provided me with some very basic understanding of some topics in biology, So I would like to share some of it with you today, go through the very basic principles of the immune system in case, like me, your formal education, your latest formal education in biology was only in high school. I think this may be helpful. So it won't be complex, but I think it's helpful if we go through it step by step. So let's start with a simple question. Well, it's not that simple a question actually, but it's simple in formulation. How does the body work? In one word. The answer to that question is proteins. Proteins are molecules that are formed of a chain of amino acids. They're the machinery of the cell. They make our body work. They're the little workers that make everything move in our body. And they're represented at the bottom of this slide as a chain of amino acids. And where do these proteins come from? They come from what makes us, us. They come from our DNA. DNA is also a long molecule, which is also a chain, but it's a chain of different things. It's a chain of nucleotides, and it becomes proteins in two stages. So you see DNA represented at the top of the slide. The two stages are transcription first and translation second. The part in the middle is what we call the RNA. It's a transportable copy of the genetic information. This too is a chain, where the nucleotides are grouped into triplets that we call codons and that correspond to the amino acids. So to recapitulate, we have DNA, a chain of nucleotides, RNA in the middle, a chain of nucleotide triplets called codons. And the end product that makes our body work is the protein, a chain of amino acids. The body puts all these molecules at work through all sorts of chemical interactions and complex processes. Let's start from DNA, the language of life. So it's a chain of nucleotides that can be of 4 types, called adenine, thymine, cytosine, and guanine. For short, we write them with letters A, T, C, G. So it's a very short alphabet, 4 letters. But when combined, this alphabet says something. It says how to make the proteins. That is, it says how to make our body work. DNA is organized into genes, and each gene has a specific function. It contains the recipe on how to make the protein. But just like kitchen recipes, each gene can have a different version. These versions are what we call alleles. The DNA lives in the center of the cell. You see a cell represented here, and the center of the cell in purple is called the nucleus. That's where the DNA gets converted to mRNA through transcription and splicing. Splicing is when the premature RNA is cut into chunks that are recombined to form the mature messenger RNA. The mature messenger RNA, mRNA for short, gets exported outside of the nucleus into the cytoplasm. And that's where it gets translated into a protein. So in the cytoplasm, we can have millions of protein molecules. But over time, these proteins can break down and they can become dangerous. So to prevent danger, the cell continually breaks down and cleans up all the proteins. What it does with them is fascinating. So the proteins are continually broken up by this machine here called the proteasome. It cuts the protein that it takes as input, if we want to use a computing language, into small chunks that we call peptides, represented here. These peptides can be captured by another special molecule called the MHC, represented in blue in the picture. The MHC then moves to the surface of the cell where it exposes the peptide that it has captured. Why does it do that? To answer this question, let's think a minute about what are peptides. So where do they come from? They are chunks of proteins. They come from proteins. That means they either come from healthy cells, or if there is something bad happening inside the cell, like it's infected by a virus or the cell is cancerous, these peptides come from non-healthy, non-self. So to recapitulate, the MHC exposes chunks that are either self or not self. So do you see where I'm going with this? At the same time as this continuous presentation happening, at the surface of the cell, we have immune cells that travel the body and scan these presented peptides. If the peptides come from an unexpected protein, they kill the cell, which provides a defense against viruses and cancer. But okay, let's imagine we are a virus or a cancer. We can find an easy trick. and indeed one that many viruses use. We can block the presentation altogether, penetrate inside the cell, and then prevent it from presenting anything to the outside world so that it doesn't get killed by immune cells. Well, no, we can't do that because there are other cells that are called natural killer cells. These ones check for these type of inactive cells and kill them. So to summarize, the cells must present peptides constantly, and they must not present unexpected peptides. Otherwise, they will be destroyed. So this was a very basic introduction to the immune system. After going through the principle, I suggest we see it in action. So we have here a video of a cancer cell, the big blue cell in the middle. And then a cell of the immune system is represented in purple. It's a killer T cell. Once it detects a cancer, it kills it. This is the defense that our bodies have against cancer. So it would be very nice if it always worked like this. So unfortunately, it doesn't. But on the plus side, that's why I'm here talking to you about AI in biology today. So why am I here? What is this all about? Why do we care about doing AI in biology? There are many reasons. I think my favorite is that we're not just trying to get another paper or make a new clever, impressive chatbot here. We're trying to change human lives. That's the sort of opportunities we are talking about. But with such an ambitious goal, we have to ask ourselves, why do we suspect this might work? So let's start by looking at one particular problem, one that concerns these machines that make our body work, the proteins. Understanding a protein can mean a lot of different things. In particular, it can mean understanding what 3D shape it will take. Indeed, the structure of a protein determines its function. Proteins with similar shapes will tend to do similar things. And we know also that the 3D structure of a protein is determined primarily by its amino acid sequence through chemical interactions. So an amino acid sequence can be represented as a series of letters. Also, in parallel to knowing this, we have seen in recent years huge breakthrough in the field of natural language processing, understanding human language. ChatGPT is a well-known example of this. These methods, these breakthroughs, rely on the huge amounts of text data that's available online, and they can be typically trained with tasks like masked language modeling. There is now also a huge amount of protein sequence data that's available online. So the reality of natural language processing is more complex than just masked language modeling. But what I want to draw attention to here is the parallel between protein sequence data and human text data. If you can train models to understand human language by asking them to predict masked words in a sentence, it is a legitimate question. Can you train such models to understand proteins by masking amino acids? So this question has actually been answered. And the answer is yes, you can. The history of protein folding is full of breakthroughs that continue until today. So a big milestone in the journey towards solving this problem was in 2020, when DeepMind released AlphaFold 2. AlphaFold 2 was a very impressive model that leveraged several innovative tricks, notably, for example, invariant point attention, just to name it, which is a special attention mechanism insensitive to, for example, 3D rotations, because when you deal with structural data, you don't want to be influenced by which angle you look at the structure from. But importantly, AlphaFold 2 was trained on supervised data. It was trained on 3D structures that were derived experimentally by people in a wet lab coat. 1 year later, however, Meta published ESN2, which showed that protein structure information could be acquired from masked language modeling only. So it was not obvious before they did it that this seemingly meaningless task would work for anything else than human language. And yet it did. Without any supervision, ESN2 acquires enough information, requires a certain amount of structural information about the protein. And then with some fine-tuning, some specialization of the model on the experimental data, it can reach a performance close to that of AlphaFold 2 on some proteins. Then more recently, we saw some new trends appearing in this field. We can mention, for example, multimodality. So AlphaFold 2 worked mainly on monomers, which are chains of single chains of amino acids. But now AlphaFold 3 deals natively with complexes of multiple proteins that interact with one another, called multimers. Also, it deals with different types of molecules. So it can model protein-ligand interactions, how a protein might interact with a DNA or an RNA molecule, for example. On the same theme of multimodality, ESM3, the successor to ESM2, also now treats more than just proteins. It treats sequences, their structures and their functions, all as discrete sequences, as sequences of discrete tokens. And it can generate sequences by conditioning on any of these simply by masking parts of the input. Finally, one trend I've noticed and that I find quite interesting is the use of synthetic data. So AlphaFold 2, for example, had self-distillation, which is when you retrain your model on its own predictions. Then in ESM3, they even use a smaller model to train the bigger, more important one. So this was just a very brief introduction and summary of the history of protein folding, but what I'm here to talk about today mainly is what we can do, and indeed what InstaDeep has done, on about DNA data. So DNA data or genomics data is very valuable because it contains information about proteins. It contains all the information in the body. So of course it's very valuable. In addition, it's getting cheaper and cheaper to acquire. Just to give you a range of ideas, in 2000, when we first sequenced the first human reference genome, the experiment cost $100 million. Nowadays, it costs less than $1,000. And this is just 25 years later. So this means we can acquire more and more genomes, even more species, and this means the amount of data available is growing every year. At the same time, we also have Moore's law, which states that compute becomes cheaper and cheaper, and therefore more powerful and more powerful. And this means that these three things combined give us the promise of an exponential progress in our understanding of genomics data through AI methodology. So it means it's only the beginning of using AI for targeted therapies, for example, or for humanity's ability to understand the genome and repress genetic diseases, for example. So InstaDeep, with this promise in mind, have developed several models that function on DNA data. It first started with human genomics data, and that led to the nucleotide transformer. Then the family grew inside the nucleotide transformers, adding more and more genomes, and then eventually also adding multiple modalities. So not just DNA, but also combining the DNA information with RNA and protein information. This was done for the isoformer, which predicts isoform transcript expression. I'll explain that later. Then I will also be talking about two other models where perhaps we can sense the reality of the applications a little bit more directly. SegmentNT is a model that can do multitasking. It annotates genome sequences with different predictions all at once. And then finally, ChatNT lets a user interact with the DNA data directly through human language interactions. So it lets a user ask questions in English, in plain English, about DNA data and gets accurate answers. So I've talked about mass language modeling, but I haven't really gone through it because I suspect many of you might know a lot about it already. Just as a reminder, mass language modeling is when you are given a sequence without any labels. So the only thing you know about your input sequence is that it exists. That's its only merit. You know that it exists, and you mask parts of it, and you ask a model to predict what you have masked. And this is the training task, the pre-training task that we talk about when we talk about mass language modeling. So the first thing I want to show about the nucleotide transformer is the power of this task. We knew it worked in NLP, but intuitively, we understand why this might work. We understand why when we hide the subject in a sentence, a model can maybe be able to mix the context and use the verb or the previous sentence to guess what the subject is. We have also seen that this has had successes in protein folding. But it's not obvious at all why that is the case. So it's fascinating to see that it also works in genomics. So the idea is that without any supervision, the model has to mix the information to understand what makes it likely or not for a certain type of, for example, amino acids or a certain type of nucleotides to be in a certain position. In the nucleotide transformer, it was found that this task let the model separate the representations of different sections of the genome. That's what's represented on the GIF on the slide. So you have, without any labels, you have the model separating, for example, the regions that code for proteins from the regions that are in between genes that do not code for a protein. So for example, in red, you have the intergenic regions, we call them. The internal representations of the model are grouped together for this type of sequences, or even though it was never told explicitly that these sequences were similar in some sense. So this is the first hint of the power of these models. So when I talk about the nucleotide transformer, I don't just talk about one model. It's actually a whole family of models that have a BERT-like architecture that were trained on a large-scale infrastructure with various sizes and on data sets of varying diversity. So the first model was trained, as I was saying earlier, only on the human reference genome, and then more genomes were added, then even multiple species. We even had a model for plants, for agricultural applications like crop design, for example. And finally, there was a version 2 of this model that was trained on multiple species with architectural and innovations as well, and where the size was also studied in detail. So one of the key findings from this family of model is that the trend that we were talking about earlier towards bigger scale, for example, in the field of NLP or in the field of protein language models, where the models become bigger and bigger, is also true in this family of genomics models. So the bigger models are the best. But what's also interesting is to note that the diversity of the pre-training data also helps the model. So the best model is the one that's trained with the most parameters on the most diverse data set, and it's the one at the right of the bar chart. It's a multi-species model. Why do we know that it's the best? We know this from testing its specialization via fine-tuning on relevant downstream tasks about DNA. So these tasks, the models are state-of-the-art on a number of them, as the table shows. And for example, these tasks let the models, these models can identify genes, understand the impact a gene variation may have on the body. For example, does it cause a disease? Does it what different things it can do? And it can also, for example, locate promoter regions, which is helpful if you want to do targeted therapies or synthetic biology. And it does many other tasks like this that can help develop precision medicine. So in short, nucleotide transformer models are powerful foundation models that understand DNA and can be specialized via fine-tuning. So after these first successes, the teams at InstaDeep took the results to the next level by adding multitasking through segmentation. This was inspired by computer vision. In the early days of computer vision, you had models that could, for example, predict if an image was of a dog or of a cat, or of a car or of a bicycle. And then as the research progressed, we saw the emergence of models that could segment all the pixels in an image and assign a label to every one of them. So, for example, on this slide, you had models that could detect the bus, the car, the bicycles, the road, whatever. So similarly, SegmentMT annotates sections of the genome efficiently and accurately, instead of having to rely on multiple specialist models. The method to do this was also inspired by computer vision. It's the well-known UNet architecture, with one key difference with computer vision models, which is that here the convolutions are in 1D, because obviously the input is a sequence, so it's a 1D input instead of a 2D input like an image. This head is added on top of the powerful pre-trained nucleotide transformer, and the whole system is fine-tuned end-to-end. So this results in a model with an extended context size that is capable of annotating genomic sequences accurately across 50,000 nucleotides. So here is an example output from segment-NT, where we can see features like genes, promoter regions, exons, introns, clearly identified and in line with the ground truth data. This model is, for example, state-of-the-art for splice site prediction. Splice sites is where the splicing of the RNA happens, and the premature RNA gets recombined into the mature messenger RNA. So we have talked about multitasking. Now I would like to talk about multimodality. So DNA contains the recipes to make proteins, and it contains all the information about a body. So here is a legitimate question. Are DNA models enough to predict protein tasks, since the DNA contains information about proteins? So the bar charts on this slide show that DNA models can do some protein tasks just as well as protein models. For some, it can even do better. However, for some tasks, it's still lagging behind. So the answer to the question is unfortunately no. DNA is not all you need, but it's a very promising first step towards multimodality. So what DNA can do is complement the protein information. From these observations, the teams decided to stack two models on top of each other. So one dealing with protein data and one dealing with DNA data. And the result is enhanced performance across the board. So the brown bar, the bar chart, shows the performance you get when you combine the two types of information, and it is above whatever you can get with only one type of information. So of course, this first result motivated looking into training directly on multiple modalities. So one way to do this was via the architecture represented here. It is a flexible approach that combines three types of modalities: DNA, RNA, and protein, that correspond to the central dogma of biology. This architecture is simple in spirit, and it's flexible. You can put your favorite model for each of the modalities. Then you aggregate the representations. For example, you can run cross-attentions, where you update the representation of each modality by cross-attending to the other two. And you fine-tune the whole thing end-to-end to obtain a model that can deal natively with all three modalities. So I will show an example of how this was applied in the next slide. And in that next slide, for example, the DNA model is the N-former, which was not developed at InstaDeep and is a powerful DNA model that can handle very long DNA sequences. The RNA model is the nucleotide transformer I was just talking about. RNA is just the working copy of the DNA, so you can use the DNA model to build representations for it. And the protein model is ESM2 that we were talking about earlier. So what is the task that this model tackles? It's RNA transcript isoform expression. That's a lot of words if you don't know these words. So a transcript is the RNA molecule that's made when a gene is read and copied during transcription. So it's basically the working copy. But there can be multiple transcripts. There can be multiple versions for the transcript that we call isoforms. Each gene has its own unique function, but the isoforms are different expressions of that function. And so DNA models can predict the expression, how much a gene is used of canonical genes, but they can't natively predict the fine-grained profile of how much each isoform is expressed. And so that's what this architecture was used to tackle. So, because of course, I should say a word about why this is an interesting task. It is interesting to know how much a gene is expressed, but it's more interesting to know how it is expressed, with what isoforms, because this might allow you to understand better certain contexts, such as cancer, how a cancer tissue differs from a healthy tissue right next to it, for example. It allows for a better understanding of the biology of the cell. So what the paper shows is that when you use the DNA model only, you have some level of performance that is decent, but that performance is enhanced as soon as you add new modalities. So as soon as you add protein information or RNA information, you get better performance. But really, the best performance is when you combine the three modalities together, and you get a state-of-the-art model for predicting isoform transcript expression. This is because the modalities complement each other, they enrich each other, they contain, they capture different parts of the information in the three modalities that correspond to the central dogma of biology. So with this success in mind, let's just add one more modality. The absolute king of modalities, the human language, English in this case. It's also useful to do that because it lets you multitask very easily. You just have to convert your data set into a set of questions and answers. So the nucleotide transformer maps DNA sequence to an internal representation. And human language models do the same for written text. So the idea is that to ask questions about DNA, we can train a model to transform the DNA representations into ones that the human language model can understand. We can project the output of the DNA model onto the input space of the human language model. The human language model used here was Llama, developed by Meta, and it was kept frozen so that it could maintain all its capabilities, like coding or just answering naturally in human language. This results in a very powerful model that lets you ask questions about the DNA. A key trick that's shown in the paper that I quite like is that if you do this, you risk having an information bottleneck on this arrow here, on the projection of the DNA information onto the space, the input space of the human language model, because you have multiple tasks possible in the questions. And so this means that the projection layer would have to project all the information needed to answer all of the different tasks if you just did that naively. So one thing that you can do, and that was done and that was proven to have better performance, is to use the question representation to modulate the projection via cross-attention. You can slightly change the projection so that it's better suited to answer the question. So this results in a very powerful model that made the cover of Nature Machine Intelligence recently, actually. And just because it's cute, I've put a few pictures on the slide of what the chatbot looks like. So you can really just ask questions about a DNA sequence, and the model answers accurately. So it's if we think about its applications, it's very useful, for example, in context where you have users that do not have a programming background. Which is extremely useful in a field where you want to work at the intersection of multiple disciplines like AI and biology. So all the models I've talked about are open source and available freely on GitHub or Hugging Face. So if you're interested in this domain, please go ahead and download them. We are very excited to see what you can build on top of this research. So now to finish this talk, I would like to present two more slides about another type of model recently introduced at InstaDeep. These models I will talk about are Bayesian flow networks, or BFN for short. So they were quite recently introduced, and at InstaDeep they were applied on protein data. So we are not talking about genomics data anymore. But before I dive into how they were applied to proteins, I think it's worth maybe going through what the models are. So Bayesian flow networks, what are these models? So I think maybe that's the only slide without any mention of biology in this presentation. So BFNs are generative models. What are generative models? Generative models are models that learn the joint distribution of the data and not the conditional probability of the labels given the data. So in this regard, they are just like encoder-decoder models, GPT architectures, for example, or diffusion models. I think probably many of you know about diffusion models very well. So how do these models work roughly? So they are complex, but I will try to briefly explain the training and inference procedures as best I can. So just like in diffusion, the first thing to note is that there is a time component. So this image represents 9 time points for a single variable in the input data. It's not 9 variables, it's just 9 time points for the same unique variable. So you have to imagine that for all the variables in your input data, say your input data is a sequence, you have 1 image per position in the sequence, and here you have 9 time points that are sampled between 0 and 1, time is treated continuously. You start with, also the other important thing to note is that the Bayesian flow networks are tasked with learning not to generate the data itself, but rather to generate the distributions over the data. So to learn the parameters of a probability distribution, not to learn the actual data itself. So you start with a prior, during training, you start with a prior distribution for each variable independently. Let's say you have 3 outcomes. Let's say it's a discrete, a variable that is discrete. So it can, let's say it's a sequence and it can be A, T, or C. You start with a prior that gives you absolutely no information about what the data looks like. So you start in the middle of that triangle. Under this prior, you can't guess anything about the data. So then you sample a noisy observation of the data. So now in diffusion models, if you're familiar with diffusion models, that's when you would ask a model to predict what noise you've added to the true observation, to the true data point. But instead, here we hold on a little bit before we start using a deep learning model. And here, because we are treating each variable independently, we can use the rules of Bayesian inference to update our beliefs on the data distribution. In other words, we update what we think the colors should be in the next triangle via the rules of Bayesian inference using the noisy observation of the data. We have done this now for all the variables independently, so for lots of images. And now that's when we can use a model, give all the beliefs about the variables, about all the, well, I say independent, they're not independent, but about all the variables independently. We give that to the model, and the model updates the beliefs. And it's the model who's tasked with mixing the information from the different variables. So the model gives us the colors in the new triangle, so in the upper middle. Once we've done this, we can now add a few more bits of information and sample a new noisy observation of that same data point. And again, with it, we update our beliefs on the data distribution. We give these beliefs to a model. The model updates these beliefs for us, and so on, and so on, and so on. And so we do this up until time 1, where we see at the end of times, we have so much information about the data that the distribution is pushed into one corner. We have no doubts anymore about what the probability distribution of that data is. It's, we said ATC, so it's a C in this case. And so that's the training procedure. And the loss that is used during this training is essentially the difference between the colors that the model outputs and what the true colors are, because with the rules of Bayesian inference, at each step, you can just compute the probability distributions based on the data points that you have sampled. Now, at inference time, you can't use for each of these triangles, you can't use a sampled noisy observation of the data, because at inference time, there is no data. You're making the data. That's what you're trying to do. So instead, what you do is that you hallucinate the data point by sampling from your own beliefs. And that's represented by the white lines in the middle of the triangles that follows the density of the probability distribution. And so you do the same communication protocol across time, and you obtain a data point at time 1. All right, so I hope I explained that relatively clearly. If I didn't, I think the one thing to remember is that the Bayesian flow networks are generative models that predict the distributions, the parameters of the probability distributions, rather than the data itself, like diffusion models would. So why are they interesting now for biology? Well, biological sequences have 2 properties that are worth remembering. First, like all sequences, well, like many sequences, they are discrete, and their left to right order does not matter. So in a human text, if you get the first word wrong, it's a problem, because if you get the subject of the sentence wrong, the whole sentence means something different. In biological sequences, the first position does not matter so much because we have such wide range interactions. We fold into a 3D shape. If your first amino acid is wrong, the rest of the protein is maybe actually very good. So the order does not matter as much as in text. So diffusion models are interesting, but they are difficult to use on discrete data because you have to add Gaussian noise, and so it's difficult to do that when you have discrete data. Autoregressive model like GPT architectures, so encoder-decoder architectures, are not ideal either because they assume a left-to-right order. So the losses are more important, the errors at the start of the sequence are more important than at the end. And so BFM models, they do not actually model the data itself, rather they learn the parameters of its probability distribution. So this makes them a perfect fit for discrete data because the probability parameters of a discrete distribution are still continuous. They sum to 1, etc. Like the probabilities are continuous. And because they treat each variable independently, as we saw, there is no left to right order assumption. For the same reasons, these models are also very useful for having multiple modalities, because whether the data is discrete or complex or whatever, it's still just modeling, we're still just modeling the parameters of its probability distribution. So let's see what this looks like when applied to biology. So I invite you to read the ProbBFN paper published in Nature Communications recently. You will see how this promising framework has been applied to protein tasks. But also what you will see is another key finding of many InstaDeep papers in biology. Data curation is key. So cool methods, clever methods like Bayesian flow networks are very promising and interesting, but just as interesting is making sure your dataset is well curated. So in ProbBFN, the dataset was carefully clustered and curated to make sure that the model only saw proteins in which our scientists had trust in. So using various metrics, they made sure that proteins for which there was a doubt of their existence, for example, were discarded. And also that there was sufficient clustering in the dataset to avoid seeing the same data points too many times. This results in a model that is capable of generating protein sequences that are simultaneously diverse, natural, and novel, even though these three things might seem contradictory at once. So what do they mean? Diverse means that the proteins generated by the model cover a wide range of protein families, of different protein families. Natural means that the sequences that are generated by the model align closely to clusters of known natural sequences from known databases. And yet they are novel because their sequence identity is not far from that of the training set. That's of course very important so that we don't just memorize the training set, because what's the point of generating novel sequences if they are already in a database somewhere? We wouldn't need deep learning for that. So for example, by further fine-tuning this model on antibody sequences, we obtain ABFN. So antibodies are another part of the immune system that target antigens, that target pathogens in the body and attract the parts of the adaptive immune system to kill it. So they are very important, and many therapies are being developed that design antibodies that target those antigens that are escaping the immune system. So it's very promising, for example, for cancer, because cancer is a cell that escapes the immune system, because if you can design an antibody that targets a patient's cancer and you inject these antibodies, you can train the patient's immune system to kill their cancer. So this fine-tuning on antibodies yields a model that is capable of zero-shot in-painting parts of the antibody sequences. This means that just by seeing antibody sequences, the model becomes able to be given parts of an antibody sequence and recover natural, novel, and diverse masks. And it does so at the same level of performance as a specialized model that had been trained explicitly on this task. So I did not show on this slide the results of the next model, which is ABBFN2, to keep it short, but I encourage you to go check out our blog or indeed the paper itself. It is quite impressive to see that you can, so ABFN2 is a model that works not only on antibody sequences, but also on many other modalities, so properties of these antibody sequences. I think it deals with up to 45 modalities, I think, from memory. And so this means that you can ask the model to generate an antibody sequence with a certain property, for example, how natural you want it to look, or how stable you want it to be, or whatever. And it's really impressive to see that the model gets satisfied requests pretty much every time. So if you're interested in that field, please go check it out. All right, so I'm now nearing the end of the presentation. So just so that this presentation would include literally all the knowledge in the universe, I thought I'd include a slide that mentions everything I had not talked about. So that gives you an idea of the immensity of what remains to be done. So there's, for example, AgroNT. We already talked about that. That's a model trained on plants for example, for things like crop design. For example, to build crops that are resistant to special diseases or the weather or whatever. Then there's also at InstaDeep InstaNovo, which is a model that helps improve the readout of wet lab experiments. And by improving the readouts, I don't mean just helping people understand it better. I mean like actually deconvolute the information that's in there, because sometimes wet lab experiments can have information that's very hard to interpret, so deep learning can help with that. We could also have talked about vision models. There are models out there that segment tissue samples, that take histology models that work on pictures of skin or whatever, of zoomed in tissues where you can detect, for example, cancers or learn about cell-to-cell interactions. And importantly, etc. There's a lot of things happening in this field. So thank you very much for listening to me. Just a final word, if I may, because I'm not actually an academic researcher, I'm an applied researcher. So I just thought I'd finish this talk with what exactly we mean by applied research, or at least what I understand applied research is. So I would say very simply that applied research is research that is closer to the real world problems, so closer to the impact. We can be, of course, the intermediary between the academic research and the actual solution, but much more than this, I think that applied researchers are tasked with identifying actual problems that need a solution, and importantly, find the solutions for them. So sometimes the solution may be just fine-tune an existing state-of-the-art model. Sometimes it will mean developing your own foundation model and testing ideas that no one has the answer to yet. I think if you're looking to work in applied research, I mean, I take the liberty to give some advice, because I suspect many of you, some of you might be considering that and have not got any experience in that, but please pardon me if I overstep here. But what I would say an important piece of advice is that often there is a lot of jargon and domain-specific knowledge, and that it's important to not hide behind the excuse of I'm not an expert. It's important to be curious, to have lots of discussions with people that come from another discipline, because by learning about the specificities of your industry, that's where you can deliver value, just simply by being curious. And that's also where you can identify problems. And so finally, I think that a bit like in software teams, I think that's crucial in applied research, is to own your product, but your product is not the software, it's the research expertise that you have developed, your skills, your team, and also your tools. So when you invest on a tool, for example, the debugging tool, like just a simple diagnostic tool, that can be useful, and it's something you have to share with your teams and the people you apply the solution to, because I've seen several times examples of just simple diagnostic tools that actually have been turned into an actual product and that change someone's job for the better. So it's not always about the coolest frontier AI research. Sometimes just a simple tool can really deliver, help advance other fields of research. And so I think that's a cool trick I would give about those of you who might be considering a career in this field. So yeah, you should just, I think in applied research, we just don't fear simple solutions, but we also don't fear complex solutions. And so what we do is we do both. And I'm very happy to be in a team where we do both, but always with impact in biology. So thank you very much. And I'm here for a few questions if you have some.